Hi everyone. I will be talking about the functional residue identification tool, or DFRI in short, an update of that tool. I don't need to convince any one of you that the recent breakthroughs due to AlphaFold, ESMFold, and RosettaFold allowed us to have access to millions of high-quality structure models. Tools such as, for instance, DeepFry show that usage of structure instead of sequence can lead to much better function prediction. The extension of DeepFry, called metagenomic DeepFry, can allows us to efficiently map sequences to predictive structures, and thanks to that, we can use this structure-based method also for sequences. But DeepFry, the original DeepFry, produces high coverage annotations, but however, they are in some cases more general as compared to homology-based methods. In my talk, I will present updated DeepFry, release 1.1, retrained on AlphaFold miniprot dataset. DeepFry paper has been published 2 years ago. The main author, Vlad Grigorievich, at the moment left science, so we decided to take over the project. Little bit of details. So you can use DeepFry in 2 modes. The first one is sequence-based. So the sequence is one-hot encoded into a feature matrix, and then the feature matrix is passed through CNN block. And as an output, you have a dense vector of predictions of Go terms. So each position of a vector gives you kind of probability of having that the input sequence has this particular Go term. And there are 4 independent neural networks for each gene ontology and EC numbers. And you can also use DeepFry on structure-based level. So the feature matrix, the sequence-based feature matrix, is it's passed through the LSTM block and then merged with the original, and then merged with the one-hot encoded feature matrix. So it works kind of like a residual network. And of course, we also utilize the structure information by constructing a contact map, and then it's used as a adjacency matrix in GCN block with weight initialization based on the output from the LSTM block. But the output is the same, so we have a vector of predictions and also 4 independent neural networks. So we decided to retrain DeepFry on a larger data set. So we used AlphaFold database of high-quality predictions, and also utilized the miniprot gene ontology annotations. And each Go term that we'd like to be predicted needs to have at least 1 experimental annotation. And then, thanks to that, we're able to get almost 3 million of structures annotated by almost 5 million of annotations. And then we decided to make a Go propagation to increase number of annotations. And thanks to that, those 3 almost 3 million of structures are annotated by almost 60 million of annotations. And we use the original DeepFry1 architecture because it's been proven to be reliable. So we didn't want to change it at the moment, at least. And now the release 1.1 is able to predict almost 8,000 of unique Go terms, which is roughly 3 times more as compared to release 1. So when it comes to inference, so if you have a structure, you can directly put it into DeepFry 1.1. But if you have a sequence, you can either use a CNN block, so this is a sequence-based prediction, or utilize metagenomic DeepFry, which is kind of homology-based method because it first searches through similar sequence using MM6 database, and then associate the closest sequence with structure from reference database that you are going to use. For instance, PDB, AlphaFold, ESM, Atlas, MIP, and anything else. And we also wanted to know how informative are our Go terms. So we computed information content that is defined like as minus log probability of having a Go term in a given reference database. And so it's not only so that DeepFry 1.1 allows us to have more Go terms, but it also allows us to have more informative Go terms, as you can see on that plot for each gene ontology. And another crucial question is what threshold we should use to have reliable predictions. So here we have kind of calibration curves. So on the y-axis we have precision, on x-axis DeepFry score, and the left plot corresponds to DeepFry 1, and the right to the updated DeepFry. So and it's been constructed based on PDB. So at least when working with PDB structures and similar distributions, we should use DeepFry score at least 0.35 or 0.5 for DeepFry 1 and 1.1 respectively. And the worst performance for the latter at a given threshold is caused by the fact that DeepFry 1 has been trained and tested on the same distribution because it's PDB, and DeepFry 1.1 has been trained on UniProt and tested on PDB. Of course, those databases are related, but they are not like, the same. We also wanted to compare both methods, and so we utilized the Microbiome Immunity Project database that comprises microbial structural models from GEBA dataset. And as you can see, indeed, we've got much more predictions in the from the version 1.1 as compared to the original one. So we also wanted to take a look at some examples. So for instance, for molecular function. So those are our function predictions for novel folds in the MIP database. So we not only have high information content, but also very high DeepFry score. We also mapped our Go terms to Coq categories. And again, we've got higher coverage for V1.1. But there is one interesting thing, that the green categories, namely information storage and processing, are predicted by, those that are predicted by DeepFry 1 are predicted as green also, but also to us cell cycle control, cell division, and chromosome partitioning, which is somehow related. It's not the same, but it's somehow related. But this mapping is not perfect because we use some not direct method of mapping Go to Cog. So another point is that we only picked one hit per structure, but we hope you are going to address those problems in future releases. Yes, and when it comes to like running diffry on some larger databases, we randomly picked 1 million of predictions, ESM atlas predictions, ESM fault predictions from Magnify. And again, we can notice that we have a high coverage across wide IC range. So information content is on the y-axis and diffry score is on the x-axis, which is pretty nice. But unfortunately, we couldn't compute information content for all predicted Go terms. So many of predictions cannot be shown on this graph. We also compared V11 to ProTNN, and on the diffry side, we use only high quality predictions, so we score beyond 0.5, and also with information content at least 3. On the ProTNN side, we excluded PFAM annotations like DUF and putative. As you can see, so in this Venn diagram, we have a comparison for uniquely annotated proteins. And as you can see, so diffry is able to predict more. And when it comes to total number of predictions, it's the same, but so on the x-axis we have information content, but it is being computed differently, so we cannot compare like each bin in the same way. And the last thing we've done so far is to we ran diffry 1.1 on UniProt. So we use this dark subset that was constructed in that paper in the lower right corner. So there was a very nice poster yesterday about that. And so we have actually, again, high coverage of Go terms across quite wide IC range. We have slightly less predictions with very high information content, but this is expected because those are simply hard targets. And so to sum up, so as we can see, diffry 1.1 provides higher coverage I mean, it provides not only more Go terms as compared to the version 1, but also they are more informative. And when we extend it with metagenomic diffry, we can actually use this tool. We can utilize this structural information also on sequence datasets, and we are trying to upgrade the tool, those tools even more, and trying and annotate the unknown. I also have a poster today, so you can reach me out later if you would like to discuss these things. And so the original diffry can be downloaded from Flatiron Institute group repository at GitHub, and the 1.1 release and metagenomic diffry can be downloaded from BioInf MCB. So at this point, I would like to thank my collaborators. So our group leader Tomasz, Witold, Piotr, Valentin, Mary, and Łukasz. And thank you very much for your attention.
Thank you, Pawel.
Thanks for the talk. So maybe I'm confused, but is this diffry set up with precision of 0.5 as your threshold? Or it's so high quality is considered things above 0.5 precision? Is that right?
Just a second. I mean, it's actually dependent on the data set you're going to use. Because if you use a different data set, for instance, from the UniProt, so the precision might be very different for a given score.
Mm-hmm.
So at least for PDB, it's recommended to use score the threshold at 0.5 in order to have precision at least 50%. Okay.
Okay. So I mean that I'm a bit skeptical if you're calling lots of new Go terms and your precision is only 0.6. Well, maybe all the new stuff is just the false positives.
Who knows?
Great work, thank you. I was wondering if you have considered participating in the critical assessment of functional annotations competition, CAFA. It's going on right now in Kaggle. Are you in there?
Not yet. But this release 1.1 is just a warm up. So we are just starting, pushing this tool forward, because we are now keeping like collecting ideas on how to make it even better. Because we'd like to upgrade, not only update but also upgrade.
Competition's over in 3 weeks. You might want to give it a go. You might win.
Thanks.
$15,000.
And I think, in fact, there's a talk tomorrow about it.
Thank you for your presentation. I just am very curious about kind of how precise these estimators are. Have you kind of looked into kind of what happens if you give these models a loss of function mutation?
This is very good question. So we haven't looked at that yet, but I think it might be very hard to incorporate this kind of functionality, but we'll try. I mean, at least we'll try. But we haven't done it yet.